Let's move on to chapter 12. In chapter 12, we start looking at evolution on a bigger scale. So in chapter 10, we were looking at changes in population. We were looking at changes in the frequency of particular traits and changes in the quantities of alleles within a population. We also talked about Charles Darwin and how his idea of natural selection and the theory of evolution has really sort of taken hold in biology and is the foundation for a lot of what we teach and do in biology. So we're going to apply that idea now to the origin of life and the way that life has diversified on planet Earth and sort of spread out into this wide variety of plants and animals and fungus and bacteria that we see today. So earlier in the semester, we probably defined life. We're studying life. Biology is the study of life. And so we needed a good working definition. But if we go all the way back to the beginning of life, there are two really important concepts that allow us to define something as living. One is the ability to replicate. And by this, I mean replicate its genetic information. Of course, organisms also have to replicate and pass on that genetic information, but the very first life form had to have the molecules in place to be able to replicate its genetic information. The other thing that's required is the ability to perform some sort of metabolic activity. Now, metabolism is all of the processes, all of the chemical reactions within your body. So as part of the first cells being alive, they had to perform the chemical reactions in order to sustain their life. So they had to be able to perform some very specific chemical processes and also to acquire nutrients and utilize them within that chemical process. Now, when we think of living things, we have more items that we put into this definition of life, but the very first molecules had to replicate and they had to be able to perform chemical reactions. So how did life on Earth arise? Well, it's mostly speculative. There's some good theories out there based on some evidence of things that people have looked at, and the idea of the origin of life has a lot of things that are possible and a lot of things that are possible but not very probable. For instance, some people have speculated that the living organisms came from outer space on a meteorite or something to that effect. And while this is possible and we may never actually know, it's probably not very likely that it happened, simply because living organisms would have to ride on that meteor, survive all the radiation and things that it's exposed to in outer space, and then land on our planet and be able to colonize it. More likely, life originated on Earth. So there are three major steps that scientists believe had to happen in order for life to emerge. The first one is the formation of biological molecules. The backbone of all of our major biological molecules, DNA, RNA, proteins, fats, carbon and hydrogen. So we had to have these carbon and hydrogen molecules available on our planet. The second phase was utilizing those carbon and hydrogen molecules as self-replicating information-containing molecules. So we had to have some sort of molecule that could contain the instructions for life and also replicate itself. And then finally, we had to have a membrane. If you think about the most basic organisms that we have on Earth, bacteria, they are genetic information contained inside a membrane. And the purpose of that membrane is to isolate that genetic material and to isolate those metabolic processes from the outside environment. So let's talk about how those steps might have come about. So before life on Earth, conditions on Earth were not very conducive to living things. There's no oxygen, which we know is required today for things to live. It was really hot. It was filled with poisonous gases. So basically, it was like living at the mouth of a volcano all over the Earth. And so you have to think about how you could get from those awful conditions to the conditions that are good for building biological molecules. And as I said, most of this is speculative. There's some good evidence for how this might have happened, but it's sort of a ever-changing hypothesis. So we think that the Earth formed about 4.5 billion years ago. A lot of people think of that point as the Big Bang. That's sort of the prevailing theory as to how this all came about. But however it actually came about 4.5 billion years ago, the Earth gradually cooled and a crust formed, and water in the atmosphere began to condense into oceans. Now, this process probably took several hundred million years. We've been able to date the oldest rocks to about 3.8 billion years ago. And then it took a little bit more time for the first evidence of life to appear. And I'll come back to that in just a few minutes. So let's talk about the conditions, though, that would have had to have come about in order for life to spring up about 3.4 billion years ago. So as I said, we need to form small biological molecules. So there's some thought that the precursor molecules, carbon and hydrogen, and the small molecules that they form were actually formed in volcanoes. And as those chemical reactions occurred in volcanoes, we get the accumulation of things like methane and ammonia and hydrogen within our atmosphere. Now, there was no oxygen. Large quantities of oxygen on Earth actually came later, and a lot later, once we see the development of plants who could pull carbon dioxide out of the atmosphere and release oxygen in its place. But how do we actually go from those carbon and hydrogen containing molecules down to things like DNA, RNA, phospholipids, fatty acids, and amino acids? Well, the theory is what Darwin called a warm little pond. And this theory is that there were small pools of water in which organic molecules eventually came together. And those organic molecules began to form the building blocks for life. So in the 1950s, a man named Stanley Miller was working as a chemist, and he wanted to attempt to replicate the conditions that he thought might be available on Earth 3.4 billion years ago or so, and to see if he could simulate an environment in which you could actually form some of those biological molecules. So he designed this apparatus, in which we have a round glass beaker here with some hot water. We've got heat bubbling up. And then he would bubble in gases in the composition that he thought were available in the atmosphere at that time. So he had different quantities of hydrogen, methane, and ammonia, and bubbled them in. The other thing that he did was simulate lightning. So he did this by applying an electrical charge to the liquid within his apparatus, and then he allowed it to cool. So this is an attempt to mimic what might be happening on Earth. You've got these bubbling pools, you've got these gases in the atmosphere, you've got volcanic activity providing heat, and then probably lightning as well. And then as things cool, they would settle in this tube. So think of this as those little settling ponds. So within a matter of days, Urey actually showed that he was able to produce a small handful of organic molecules, specifically at least 5 amino acids. Now this is where we get that idea of primordial soup, if you're familiar with that concept, that all of these molecules mixed together in a pool of hot water and sort of formed these early biological molecules. So at this point, we have evidence that it's at least possible to make the molecules important for life out of non-living or non-biological molecules. Now, since then, the quantities and the mixture and the types of gases that were likely on Earth at the time has been revised. And so it's possible that Urey wasn't using the sort of quote unquote real conditions, but he was actually able to show that you could get something with at least some of the gases that were available. So he might have not got it completely right, but it doesn't mean that this is unimportant. They're a promising first step to suggest that complex organic compounds could have been produced from inorganic chemicals on primitive Earth. So in order for these molecules to come together into something living, we not only have to have the important biological molecule precursors, but we also specifically need to have self-replicating information-containing molecules. Now, if you remember, we've talked about this type of molecule before. Information-containing molecule, those are our nucleic acids, our DNA and our RNA. And DNA can replicate itself. And so we need to somehow get these molecules into self-replicating information-containing molecules. Researchers also believe that that molecule has to have catalytic properties. Now, a catalyst is an agent that causes a chemical reaction to go. In the biological world, these are enzymes. They are proteins that help chemical reactions along. So there's not a good hypothesis as to how we get from that warm little pond with some biological molecules to actually having self-replicating molecules. But scientists believe that you have to get to that self-replicating molecule. So the hypothesis is that the first sort of quote-unquote genetic information containing molecule is actually RNA. And this leads to a hypothesis of something called the RNA world, in which the genetic information on Earth was all RNA. And the reason for this is that not only is RNA information containing and nucleic acids are self-replicating, but some types of RNA can also work as catalysts. They can also produce or help chemical reactions along. Now, these are molecules called ribozymes. Ribo meaning ribosome. We saw that in one of our earlier chapters. Those are the molecules that help build proteins. But those molecules are made of specific RNA molecules and proteins together to make one big enzyme that is in our class was building amino acid chains and building proteins. So they believe that this molecule, this RNA molecule, not only could contain information, but could also, in some forms, actually perform chemical reactions. So we're getting close to being considered living. We've got our first part of our definition of early life, and that is that we now have a molecule that can replicate and can serve to carry out the replication process. So this leads us to phase 3. So if you think about bacteria, the two big things that they have or are genetic material, and they have a cell membrane. Now, we defined the smallest living organizational unit as a cell. And all cells, no matter what type of cell you are, have a cell membrane. So that's the next thing that we need. The reason that we need this is that membranes isolate the cell from the surrounding environment and produce a semipermeable barrier. Now remember, a few chapters ago we talked about things moving in and out of the cell. We need our membrane to be semipermeable. We also need our membrane because when we isolate the contents of the cell from the rest of the environment, we also make possible the ability to perform chemical reactions because we can have different chemical concentrations inside and outside the cell. And so if we can get those self-replicating molecules into a membrane, we have our early life form. Now the thought on this is that these early membranes were made by fatty acids. Now fatty acids are molecules that are really similar to our phospholipids. If you remember, our phospholipids had that phosphate head and the fatty acid tail, and they would self-assemble with their heads together and their tails together. I'm sure you guys remember that double membrane that we talked about. I believe it was in chapter 4 when we talked about our membranes. And so fatty acids will actually do this. If you put a fat, and you can think about this, if you put a drop of fat or oil into a bowl of water, what's it do? It makes a nice little bubble. And this is what they think happened with the early cells, is fatty acids came together and formed what we call a micelle. Now a micelle is just sort of a bubble of fat, and inside that bubble would have been our self-replicating molecules. So however we got there, our first evidence of life was a prokaryote type organism. Now we think that this guy, the fossils that we have of these organisms are about 3.4 billion years old. And this is dated by dating the age of the rock that the fossils are found in. The reason that we think that they're prokaryotes is based on their shape and the size of the impression, meaning that the shape and the size correlate to present-day bacteria. They're the right size, they look to be the right shape, and so it's a good indication that this is possibly a prokaryote. The third way that we know that these are probably fossils of prokaryotes is based on the ratios of different types of carbon within that organism. So all organisms are made up mostly of carbon. Now carbon and all other molecules will go through sort of a decay process, and these carbon molecules will do this at a very specific rate. And so if you look at the ratio of carbon-12, which is the form of carbon that living things are made out of, compared to how much that is decayed into carbon-13, you can actually use that rate to calculate the age of the organisms. And so when they did that, they found that the age of the rock and the age of the fossilized organisms were about the same. And so they were able to put what they think is a living organism with a similar date to the age of the rock, which allowed them to deduce that they think the first living organisms on Earth appeared around 3.4 billion years ago. Now, of course, they could have appeared a little bit sooner, and we just don't have fossil evidence. Remember, we talked about fossils in chapter 10, and only some things will actually fossilize, and we don't get a good representation of everything. But we at least have some evidence that around 3.4 billion years ago, life on Earth started.